Hello and welcome back everyone. My name is Shirsha, and for those of you who are new to this channel, in this channel we basically talk about the various techniques that we use commonly in our day-to-day laboratory experiments. We talk about their basic principles and what are the utilities of each of these techniques and so on and so forth. In this particular lecture, we are going to talk about DNA isolation. Now, DNA isolation is one of the most basic things that you would be performing as an experimentalist or as a researcher. And no matter whether you would be dealing with purification of proteins or you want to do some assays in cell-based systems, the first and foremost requirement for anything is the DNA. What do you specifically require? You require a plasmid or a vector which is expressing or which contains your gene of interest. So that when you put this plasmid in any expression system, whether it be bacteria or insects or mammalian expression system, this expression system will now express your gene of interest, and now you can go ahead by doing whatever experiments you want. So it is very important to know how to isolate the DNA because it is the basic starting material with which or on which you will build all of your experiments. So in this specific lecture, we'll be talking about a step-by-step procedure of how you isolate the DNA without using a kit. In most laboratories, if you're already working, you would see that mostly you isolate DNA using kits, but these kits also utilize the same basic principles that we are going to discuss in the specific lecture. So I thought that it would be a good idea if you are well versed with what is the principle behind the various steps that you're performing in DNA isolation and how it is actually done. It is easy doing something utilizing a kit, but it is always good to know what the actual steps are so that in case you face any problems when utilizing the kit, you can troubleshoot it. So without further ado, let us dive into the topic of DNA isolation. So I would like to begin with a very basic introduction. So what is DNA extraction? DNA extraction or DNA isolation is a procedure to collect DNA for subsequent molecular or forensic analysis. So all of the applications that I talked about of expressing your gene of interest in an expression system, these all pertain to molecular applications. DNA extraction is also important for forensic analysis because if you watch any of these crime shows, then you would see that they typically try to collect blood samples from the site of the crime, because then from the blood you can isolate the DNA, and from that isolated DNA you can do DNA fingerprinting, which can help you to identify who your criminal or convict or the responsible person can be. So DNA extraction is also very important for forensic analysis. The first isolation of DNA or deoxyribonucleic acid was done in the year 1869 by Friedrich Miescher. Currently, it is a routine procedure in both molecular biology as well as in forensic analysis. The separation of DNA from cellular components can be divided into three main stages. So the DNA would be present inside the cells. So how do you isolate the DNA from within the cells? So there are three main stages. Disruption and lysis of the cells. So just like in protein purification, you have to lyse or break open the cells so that you can get access to whatever proteins are present inside the cell. Similarly, for isolating the DNA, you first have to break through the first barrier, which is the cell wall or the cell membrane, depending on what cell type you're isolating the DNA from. Because once you break it through, you will now have access to the contents present within the cell. Now your DNA, if you're isolating it from a eukaryotic system, would be present within the nucleus. So you have to break through the nuclear membrane as well. But what we are mostly going to talk about is isolation of DNA from bacterial systems. So for that case, you don't really have any nuclear membrane, or you don't have to deal with one because it's a prokaryotic system. Once you've open the cells, you have to remove all of the proteins and other contaminants which would be otherwise interfering with your isolation procedure. And finally, once you have removed all contaminants, you will recover the DNA. So let us now take a look at a step-by-step procedure for DNA isolation. So before diving into the step-by-step procedure, I would like to tell you that the protocol that we are discussing over here is for the isolation of genomic DNA from eukaryotic cells. It can be either animal cells or plant cells. So we will be talking from the perspective of isolating genomic DNA from animal cells, but we can also isolate genomic DNA from plant cell using similar techniques. Only the first step, that is the step where we have to lyse open the cells, will vary. Other than that, we can also isolate the DNA from bacteria, but for isolation from bacteria, the steps would slightly vary. And if in context to the introduction that I gave you of how it is relevant for molecular biology, or how DNA isolation is relevant in terms of molecular biology, wherein we want to isolate our gene of interest which is cloned into a plasmid vector from bacteria. So that will fall under the wing of plasmid isolation. So the steps in plasmid isolation are slightly different from the steps in genomic DNA isolation. And here I would once again like to reiterate, we will be talking about genomic DNA isolation. So the first step is the extraction of DNA from cells. Cells are collected from culture flasks. So if you are culturing animal cells, then you would be culturing them in certain culture flasks. This is something that we have already discussed in depth and in detail in my lecture on mammalian cell culture techniques. So once you collect the cells, you will centrifuge it to pellet down the cells and wash it twice in phosphate buffered saline or PBS by centrifugation. Now, why is this washing step required? This washing step is required to remove any trace amount of media that might be entrapped within your cell pellet, because the different components present in media will interfere with your subsequent steps of DNA isolation or DNA purification. So for that, washing becomes important. What do we mean by washing by centrifugation? So once you have collected the cells by centrifuging, that is, you have the cell pellet at hand, you resuspend the cell pellet in PBS and you centrifuge it again. You resuspend the pellet again and you centrifuge it again. So this will help you to wash the cell pellet and get rid of any media that is trapped within it or within the cells. So you can also go to perform DNA extraction from tissues. Under such situation, the tissue is cut in small pieces, taken in tubes containing PBS and centrifuged. Their purpose being the same, where PBS will wash away the material that is present surrounding the tissue. So let us consider that you are taking the tissue from any, say for instance, it's liver tissue. So you have a liver piece and you're cutting the liver piece out of a chunk of liver. So any surrounding body fluids that would be present, that would be removed by this washing, and the washing would also help in removing any other contaminants that might be coming. So for instance, it will remove some of the extracellular matrix components as well. But again, that is something you have to keep in mind, that you will be having extracellular matrix when you're present in the tissue. So you have to take care of removing the ECM components as well. Most of the ECM components, because they are proteins also by themselves or proteins in nature, so they would be taken care of in the subsequent steps where we try to remove any contaminating proteins from the DNA isolation step. But the entire point of washing is that any bodily fluids that you have present surrounding the tissue, you can remove it when you wash it with PBS. So the pellet is washed twice in PBS by centrifugation. That is it. So the next step is washing the pellet. So this washing is different from this washing. This washing was done to get rid of PBS. And in this washing, in the second step of washing, you will be resuspending the pellet in a specific buffer or the DNA buffer. The pellet is resuspended in DNA buffer which contains tris HCl and EDTA and is prepared in distilled water, has a pH of 8, and centrifuge at 10 degrees centigrade at 2000 rpm. So this is a still lower temperature, 10 degree. The rest of the steps would all be performed at room temperature. So the supernatant is then discarded. Note, EDTA added will chelate the divalent cations, for example magnesium ions, which is necessary for DNase activity, therefore inactivating DNases and preventing DNA degradation during the subsequent steps of purification. The third step is cell lysis. So in this step, you essentially wash the pellet once in your DNA buffer to bring the cells in this specific environment of tris HCl and EDTA. The pellet is resuspended in DNA buffer. Once again, it's the same constituents. Proteinase K and 10% SDS are added, and the mixture is then shaken gently and incubated overnight at 37 to 45 degrees centigrade. So this temperature range of 37 to 45 degrees centigrade is the temperature where proteinase K exhibits its ideal or highest activity. What proteinase will do? It will degrade the various proteins present in your sample. So that will help in their removal in the further steps. 10% SDS is also added. So that is why all of the subsequent steps are performed at room temperature, because SDS would crystallize if the steps are performed at 4 degrees centigrade. And SDS will also help in denaturing the proteins. So SDS acts as a detergent, which also will rupture the nuclear membrane and help in denaturation of the proteins. And proteinase K added will also degrade the proteins. So next we will have to precipitate the proteins present. So you have degraded and denatured it. Now you have to precipitate it. So an equal volume of phenol is added. The mixture is shaken at room temperature and centrifuged at room temperature at 5000 rpm. So note, what is the role of phenol? Phenol will cause the protein to precipitate out of the solution. Saturated phenol, if added, it will degrade the DNA. So this is undesirable for us because ultimately we want to isolate the DNA. At alkaline pH, DNA will separate out into the aqueous phase, and phenol is an organic compound as we know, so that will form the organic phase. So you have to maintain the pH of the phenol that you're adding to ensure that the DNA separates out into the aqueous phase, and centrifugation will then help in separation of the aqueous and the phenolic phases. So your protein which has been degraded will come down in the phenol, which is in the organic or phenolic phase, and your DNA will stay in the aqueous phase. So the aqueous supernatant, which contains your DNA of interest, is now taken into a new tube. An equal volume of phenol and chloroform and isoamyl alcohol. So the ratio between these three would be 25:24:1. So equal volume of phenol and chloroform isoamyl alcohol. There is a total amount equal to the volume of the supernatant. So whatever is the volume of a supernatant, say you have X microliters of your supernatant. So to that you add X microliters of this mixture in this specific ratio. You add it, you shake it for 10 minutes at room temperature and centrifuge at room temperature for another 10 minutes at 5000 rpm. Now do keep in mind that all of these steps of centrifugation and mixing are done at room temperature. We are not doing any of them at 4 degrees centigrade because if you remember, you added SDS to the mixture in the first step. And SDS, if you spin at 4 degrees C, will crystallize. It will form crystals. So that is again undesirable. So all of the steps are carried out at room temperature. Note, chloroform will also cause denaturation of the proteins. It will remove the polysaccharides and lipids, all other contaminants, and stabilize the two layers, the aqueous layer and the phenolic layer. Isoamyl alcohol prevents the tanning that is caused by esterification of lipids. So again, you don't want this because this will interfere with your DNA isolation procedure. The next step is to remove the phenol now because this contains your degraded proteins and you want to get rid of it. So how do you do that? The aqueous supernatant is once again taken in a fresh new tube. An equal volume of chloroform isomyl alcohol in the ratio of 24:1 is added. So if you have X microliters of this aqueous supernatant, to this you will add X microliters of this chloroform isomyl alcohol mixture. Shake for 10 minutes at room temperature and again centrifuge at room temperature for 10 minutes at 5000 rpm. Note, the chloroform that we are adding will help in removing the phenol. So this is what it typically looks like once you've performed this phenol chloroform isomyl alcohol step. So you have the aqueous phase on top, which contains your DNA and RNA. You have this interface formed between the aqueous phase and the organic phase, which contains your degraded protein. And then you have the organic phase, i.e. phenol. So the next step is precipitation of the DNA. So now that you have removed proteins and other lipids and co-contaminants from the solution, and you have just the DNA in the aqueous phase, you now have to collect this DNA. So you will precipitate it out of the solution. So how is that achieved? So the aqueous supernatant is now taken in a new tube and the volume of it is measured. 1/10 volume of 3 molar sodium or ammonium acetate at a pH 5.2 is added, and thrice the volume of water-free isopropanol or ethanol is added. Now you can add either of these sodium acetate salt or ammonium acetate, and you can add either isopropanol or ethanol. And personally, I feel that isopropanol precipitation is better than ethanol precipitation, but that is my own opinion. Again, the mixture is shaken gently until the DNA precipitates out. And at this step, you can visualize the DNA precipitating out of solution. So note, DNA precipitation in this step is due to the ethanol or isopropanol interacting with the water such that fewer water molecules are available to dissolve the DNA molecules. So essentially what the alcohol does is that it removes the water molecules that are present in the vicinity of the DNA molecule. So now the DNA molecule is no longer solvated and it will come out of solution. So this concept is kind of similar to salting out of protein that we have discussed in one of our previous lectures on protein purification part 1, part 2 of about ammonium sulfate precipitation. So over there what we were doing is that we were removing the water molecules present in the vicinity of the protein molecule by adding ammonium sulfate. Similarly over here we are removing the water molecules from the vicinity of the DNA molecule by adding the alcohol, and this will cause it to come out of solution. Now sodium and ammonium acetate increases the salt concentration, and it essentially plays the same role as ammonium sulfate in salting out in case of proteins, and this helps in precipitation of DNA by salting out. So this term is the same, it is the same principle, salting out. So if you remember what we have discussed, or for the new people who have not yet gone through that particular lecture, I will repeat what exactly happens over there. So when you're adding the salt, in this case sodium or ammonium acetate, so sodium or ammonium ions are positively charged and DNA molecules, as we all know, are negatively charged. So what happens is the negatively charged DNA backbone will interact with these positive ions. So as you increase the concentration of the positive ion, they will compete with the water molecules that were present in the vicinity of the DNA molecule, and they will remove out the water molecule. Why? Because the interaction between the negatively charged DNA and the positively charged cation or ion, there is an electrostatic interaction, and electrostatic interaction is stronger than the interaction between the DNA molecule and water, which was based on hydrogen bonding. So it will displace out the water molecules from the vicinity of the DNA molecule, helping to bring the DNA out of solution or unsolvating it. Isopropanol can be used instead of ethanol. The precipitation efficiency for isopropanol is higher. However, isopropanol is less volatile than ethanol and needs more time to air dry in the final step. So in the final step, we have to remove the ethanol or whatever alcohol we are adding from the precipitated DNA, because once we precipitate the DNA out, we will finally want to dissolve it in a solution of interest. So to allow for this dissolution, all of the alcohol and any trace amount of alcohol has to be removed. So because isopropanol is less volatile than ethanol, so it requires more time to air dry, and you have to allow all traces of isopropanol to evaporate out of the DNA before you dissolve it. So this is what precipitated DNA actually looks like. You can clearly see these white strands and this white network sort of strands, and this is actually your DNA molecule. So what we are seeing over here, I wouldn't say DNA molecule, it's DNA molecules. So what we are seeing over here is genomic DNA, which is being isolated from animal cells. So this is what the picture is of. So once you have precipitated the DNA out of solution, it brings us to our next step, which is transferring the precipitated DNA. So for that, a sterile glass pipette is used to transfer the precipitated DNA into a tube containing 70% ethanol. So the tube is then inverted to thoroughly rinse the DNA. That is, you basically hold the tube in your fingers and you just invert it a couple of times to rinse the DNA with ethanol. And the samples are then centrifuged for 20 minutes at 14,000 rpm, again at room temperature. No, a certain amount of salt is precipitated in a bound form with the DNA, because as we discussed in salting out, because the positively charged ions will be interacting with the negative DNA backbone. So when DNA is precipitating out of solution, a certain amount of salts are also precipitated out along with the DNA. So 70% ethanol allows these salts to be dissolved in water. So that is why we do not add absolute ethanol. We add 70% ethanol, because this solution will have 30% of water, and in this 30% water, these salts will be dissolved. So the next step is drying and storing the DNA. Before going into that, I missed one point. So this particular step where you use a glass pipette to transfer those DNA strands, as we saw in the previous slide on the image, or on the previous slide in that image, from onto a fresh tube, is called spooling of the DNA. And it has to be done with utmost care. You have to ensure that while you're doing it, you don't really end up breaking the DNA strands too much. So the next step is drying and storing. The DNA pellet is then dried in a lyophilizer to obtain purified DNA. So this drying is done to remove all trace amounts of the alcohol. For obtaining even purer DNA, the pellet is suspended in an appropriate buffer and treated with RNAs to remove contaminating RNA. So if you recall, when we were talking about phenol chloroform isomyl alcohol precipitation, so we said that the aqueous phase along with DNA also contains RNA. So if you want to get rid of the RNA and you want super pure DNA, then you can add RNases at this step to get rid of any contaminating RNA. Now once you remove the RNA, steps 4A through 7, that is the entire step of precipitation, is once again repeated. Because what is RNase? RNase is also an enzyme. So enzyme means it is a protein molecule. So that also has to be removed. So you can repeat these entire steps from 4A to 7. And now you finally have pure genomic DNA which you have isolated from your cells. So now that you have purified the DNA, the next and most obvious step is to visualize the isolated DNA. Because unless and until you can visualize it, how do you know that what you have gotten is the right thing? So for DNA visualization, what we typically do is we run an agarose gel. So in this particular section of this lecture, we are going to talk about agarose gel electrophoresis, which is again, it is one of the most common techniques that are performed or carried out in laboratories, and it goes hand in hand with DNA isolation and purification. We'll be talking about the underlying principle and what are the various factors that affect this procedure. So let us start our discussion without much further ado. Agarose gel electrophoresis. So agarose gel electrophoresis is a method of electrophoresis used in biochemistry, molecular biology, genetics, and clinical chemistry to separate a mixed population of macromolecules such as DNA. Agarose gel is relatively easy to cast. It has relatively fewer charge groups and is particularly suitable for separating DNA of size range most often encountered in laboratories, which accounts for the popularity of its use. So, electrophoresis means the movement of molecules under the application of an electric field. We have on this specific channel lectured I'm sorry. We have covered lectures on SD's page, which is polyacrylamide gel electrophoresis, where we talked about separating protein molecules. So here, agarose gel electrophoresis is used to separate DNA molecules. Now, the separated DNA may be viewed with stain, most commonly under UV light, and the DNA fragments can then be extracted from the gel for subsequent use if required with relative ease. Most agarose gels used are between 0.7 to 2% in their concentration of agarose added, and they dissolved in a suitable electrophoresis buffer. So this is what a solidified agarose gel looks like. Now this three-dimensional structure is held in place together with hydrogen bonds, and therefore it can be disrupted by heating back to a liquid state. So when casting agarose gels, we typically use agarose powder. We dissolve it in the specific buffer. The most commonly used buffer is TAE buffer, the tris acetate EDTA buffer. And we then let it cool down. And as it cools down, hydrogen bonds are formed between the agarose molecules, which leads to its solidification. So if you take this solidified gel and if you reheat it, because these hydrogen bonds which are holding it in place in the first place would be broken, so this will cause this gel to melt once again. So if you have any unused gel typically, then that can, well, you can reuse this cast gel by heating it and recasting it, provided you have not added ethidium bromide, which is the common stain used for visualization under UV light, because heating would degrade the ethidium bromide. If you say, for instance, let us consider a situation that you forgot to add ethidium bromide and you have cast the gel. So instead of throwing out the gel, considering that it is completely useless, you can simply heat it up again, which will melt it. And now you can add the ethidium bromide to the molten solution and cast the gel once again. So how does agarose gel electrophoresis work? DNA is an organic acid and it is inherently negatively charged. So when the DNA is exposed to an electric field, the particles will migrate towards the positively charged electrode, that is, they will migrate towards the cathode. Smaller pieces of DNA can travel further in a given time than larger pieces. So if we are to draw comparisons with PAGE or polyacrylamide gel electrophoresis, over there we had to coat the proteins with SDS, which would impart them with a negative uniform charge, which will now allow their migration towards the positive electrode when placed in an electric field. However, because DNA is naturally negatively charged, so no pre-treatment of the DNA sample is required to ensure its migration towards a positive electron. It will inherently, owing to its physical properties, migrate towards the cathode. Also, smaller pieces of DNA can travel further. Why? Because they will face lesser resistance when migrating through the gel, whereas larger pieces of DNA or DNA which is more spread out, that is, it is occupying more volume, will face more resistance as it migrates through the gel, and this will retard down its migration, causing it to travel shorter distances compared to smaller pieces of DNA or DNA which has lesser amount of volume. So what exactly do I mean by DNA with more volume or less volume? Say you're running plasmid DNA on a gel. So the plasmid DNA can either exist in a normal circular form or it can be supercoiled. So because the supercoiled DNA or the supercoiled plasmid will be occupying less volume, it will be more compact in its size. So that will ensure that it migrates further because it will face lesser resistance when migrating through the agarose gel. Whereas the circular form of the DNA, the circular plasmid, although it is of the same size as the supercoiled DNA, it will, because it is more spread out, it will therefore face greater resistance when migrating through the gel and will migrate shorter distances. So yes, once you have cast the gel, you typically load your DNA samples onto the wells, and for loading you have to add certain specific loading dye. The loading dye will do nothing but it will weigh down your samples. It will typically have glycerol and it will have a stain. So the stain will help in visualizing the dye front as the gel is running, and the glycerol present will weigh down your samples. So when you pipette the sample into these wells, it will ensure that instead of going everywhere, the sample settles down into the well. So the loading dye will again have certain roles that are common with the loading dye which is used in polyacrylamide gel electrophoresis. Next, you apply the current. So when you apply current, you see that the wells are present towards a negatively charged electrode and DNA will migrate towards a positively charged electrode. So you have to be very careful. If you place it in the opposite direction, then all of your DNA will go directly from the well out of the gel. It will just migrate from the well to the top side, and since there isn't much agarose gel over there, it will simply migrate out of the gel and into solution. And once your gel has run, you can analyze the gel. The particles would be separated out, or the DNA molecules, the DNA rather, would separate out on the basis of its size. So how do we exactly cast an agarose gel? So pre-clean the gel casting plate to remove any chunks of agarose that might be present from the previous gel that was cast, and securely place it in the casting apparatus. So you have to place it securely to ensure that when you pour in your gel, the liquid form of the gel, it doesn't really leak out of the casting plate. So for that you have to securely place it. Weigh the appropriate amount of agarose and transfer the agarose into a conical flask. Add the appropriate amount of gel running buffer, that is TAE, tris acetate EDTA buffer. So typically your tris acetate EDTA buffer contains 40 millimolar of tris, 20 millimolar of acetic acid and 1 millimolar of EDTA. Boil the mixture to dissolve the agarose. Add ethidium bromide, mix it in by quickly just swirling the conical flask, and quickly pour the mixture into the gel casting plate. Like I previously mentioned, if you forget to add ethidium bromide at this step and you just ended up directly pouring the mixture into the gel casting plate and your gel has now solidified, so instead of throwing away the gel, you can simply cut it into smaller pieces, place it back into the conical flask and then heat it up again, because heating will cause the breaking of the hydrogen bonds that was actually holding the gel in place, and now it will once again turn into a liquid, and you can add the ethidium bromide, mix it and pour it again into the gel casting plate. Insert the comb carefully, ensuring that there are no bubbles, and you then let the gel solidify. Now one critical difference between the SDS page gel and this agarose gel is that SDS gels or the protein gels, let us be more generic, protein gels, they always run in a vertical fashion, whereas agarose gels are always horizontal. So the gel would be horizontally placed and it will run in a horizontal fashion. So this is what a gel casting and running apparatus looks like. So you see, this is your casting plate and these are the blue ones are the combs, and you have these white apparatus that is for actually holding this gel casting plate in place. So it will seal the two ends because the top side and the bottom side are open. So it will seal them so that when you add the mixture into the casting plate, it doesn't leak out. And this is your apparatus. So you see, this is a horizontal gel running apparatus. So now we will briefly talk about the various factors that affect the migration of DNA through an agarose gel. So a number of factors can affect the migration of nucleic acids. Number one, the dimension of the gel pores, which in turn is determined by the gel concentration. So a linear DNA fragment of a given size will migrate at different rates through gels containing different concentrations of agarose. There is a linear relationship between the logarithm of the electrophoretic mobility of the DNA, that is the ability of the DNA to migrate under the application of an electric field, and the gel concentration. So most commonly cast gel agarose gels are of 1% concentration. As you increase the concentration of agarose, this is again very much similar to polyacrylamide gels that are used for protein visualization, where when you increase the concentration of polyacrylamide, the gel becomes more and more dense, in the sense that the size of the pores present in the gel becomes smaller and smaller. Similarly, as you increase the concentration of agarose, the size of the pores present in the agarose gel become smaller and smaller, and smaller pores will actually lead to greater hindrance in the migration of DNA molecule. So if you have larger pores, then the DNA molecules can easily insert into the pores and migrate through the gel while facing lesser amount of resistance. But as you will reduce the size of the pores when you as you will let me restate that. So as you increase the concentration of agarose, the size of the pores will decrease, and as the size of the pores decrease, so the hindrance to migration of the DNA molecule would be greater, and this will slow down the migration rate of the DNA molecule. And in fact, the relationship between the electrophoretic mobility of the DNA and the gel concentration is very linear. So as you increase the concentration of the gel, the electrophoretic mobility goes down. So the relationship is inverse. So typically, this is a table that will give you an idea of what concentration of agarose gel should you cast depending on what is the size of the DNA that you want to resolve. So resolution refers to the ability of the gel to separate out two molecules within this specific size range. So if you want to separate molecules of which has a size range of 1000 to 25,000 base pairs, then 0.5% agarose gel is best suited. Similarly, as the size of the molecules that you want to separate out decreases, the concentration of the gel that you will be using increases. So for instance, when you want to separate out DNA molecules having a size of 50 to 1500 base pairs, you use 2% agarose gel. And this is an example showing how the concentration of the gel can affect the electrophoretic mobility of the DNA. So this band or this DNA band is the same as this DNA band, but you see, as you're increasing the concentration of the gel, so the electrophoretic mobility is reducing. That is, the distance migrated by the band or by the same band of DNA, that is DNA molecule of the same size, is lesser and lesser. So as you increase the concentration, the migration is retarded and the DNA band will now be obtained at a higher position. Okay. So the next factor that we are going to discuss is the size of DNA being electrophoresed. So the size of DNA is also equally important in determining how far it would migrate. So molecules of double-stranded DNA migrate through the gel matrix at rates that are inversely proportional to the logarithm of the number of base pairs. Larger molecules will migrate more slowly because greater frictional drag will be faced by them, and because they worm their way through the pores of the gel less efficiently than smaller molecules. So what happens is that the smaller molecules can easily enter into the pores present in the gel, and therefore they can migrate easily and more quickly. As compared to that, the larger molecules, because of their larger size, cannot really insert into the pores of the gel so easily and so efficiently, and this will retard their migration because now they'll be facing greater resistance. So typically the relationship between the distance migrated and the size of the DNA fragment is an inverse relationship. And as you increase the size of the fragment, it will migrate lesser and lesser distance. You see this one, which is of the highest size, migrates the least, and then as the size is decreasing, the migration distance covered is more. And this relationship is this linear relationship is actually observed between the distance and the log of the number of base pairs. Okay, so if you take a look at this, you see the shortest DNA molecule has migrated the furthest, whereas as you go up, you have DNA molecules of progressively larger size or length. The next factor that we'll talk about is conformation of the DNA. Now, the conformation of the DNA will also affect its migration ability. So, superhelical circular form 1, Nick circular form 2, and linear form 3 DNAs migrate through the agarose gels at different rates. So, as I have briefly mentioned in one of the previous slides, that circular plasmid DNA can exist in multiple forms. It can either be in the linear form. So why is it linear? Because during the isolation procedure, some of the DNA molecules, they got nicked on both the strands because this is double-stranded DNA, and now it has become linear. You will have nicked circular. So because there is a nick in it, it will, but the nick is just in one of the strands and in not both the strands. So this will cause uncoiling of the supercoiled DNA, and it will be simply circular. And you have the supercoiled superhelical circular form. And each of them, because of their compactness, will migrate at different rates on an agarose gel. So the relative mobilities of these three forms depends primarily on the concentration and type of agarose, the ionic strength of the buffer used, and the density of superhelical twists in form 1 DNA. More dense the twists are, more compact the DNA molecule would be in its size, and further it will migrate. So this is what it would look like if you run uncut or undigested DNA. Undigested in the sense that you have not treated the DNA with any restriction endonuclease. So you will have your superhelical form, which being the most compact will migrate the furthest. You will have the nicked circular form, and finally you will have the linear form. But I would like to state over here that with the kits available for plasmid DNA isolation these days, these kits are really efficient, and typically this population is not obtained when you isolate plasmid DNA using the kits available these days. And when you digest the DNA with some restriction endonuclease, the band you will obtain only a single band, which will be of a higher size. Obviously, higher size compared to the superhelical or the nick DNA. It will be at the same length as the linear DNA over here. Okay, so if you compare uncut DNA and cut DNA side by side, and provided that digesting the DNA did not really lead to release of any DNA fragment. So let me restate that. So typically you digest the DNA to remove your gene of interest from the DNA plasmid, right? So let us assume that you digested the DNA such that the enzyme is only cleaving the DNA at one side, but it is cleaving both the strands. So it is a single enzyme digestion, and you have digested such that there is only one side in the entire DNA molecule where your enzyme will go and cleave. If the enzyme was cleaving in multiple sites in the DNA molecule, then you will obtain multiple fragments over here depending upon the size of each of these fragments. So if the enzyme cuts only once in the entire DNA molecule, so this cut DNA will typically migrate slower than the nicked DNA. it will migrate at the same level as this linear DNA that you will be getting in the uncut population or uncut DNA population. But yeah. The rate of DNA migration is also affected by the voltage that is used. So at low voltages, the rate of migration of linear DNA fragments is proportional to the voltage applied. However, as the strength of the electric field is raised, the mobility of the high molecular fragments increases differentially. So greater the voltage, faster would be the migration. But you want to run your agarose gels at an optimal voltage so that your DNA molecules are not really rushed through the gel, because if they're rushed through the gel, then you're not really allowing them sufficient time to resolve properly. So to obtain best resolution, DNA gel should be run at low voltages. So typically it is run at 100 volts. If you're casting 1% agarose gel and running it, you will typically run it at 100 volts. And the voltage used may vary depending upon the concentration of the agarose gel that you're using. The ionic strength of the buffer used also affects the electrophoretic mobility of the DNA, and so does the concentration of the intercalating dye such as ethidium bromide if used during electrophoresis. So this is how a DNA gel looks when visualized on a transilluminator. So a little bit about ethidium bromide, since we have mentioned it so many times, and that is what we are using to visualize the gel. So what ethidium bromide does is that it will intercalate in between the base pairs of the DNA molecule. Now, when it intercalates over there, so ethidium bromide is present everywhere in the gel because you are adding ETBR to the gel mixture before you are pouring the gel into the gel casting plate. So it is present everywhere. When any DNA molecule is present, this ETBR in this specific region will intercalate in between the base pairs of the DNA molecule. Now, as it intercalates over there, the environment faced by it is hydrophobic when intercalated between subsequent base pairs, and in the hydrophobic environment it will fluoresce. So it will light up only in those regions where the DNA is present and not elsewhere, although note it is present everywhere in the gel. And this is what the DNA looks like when visualized using a chemidog. So over here also the stain used is ethidium bromide. These are just two different methods of visualizing the gel. One is using a transilluminator and one is using a chemidog system. Now this using a transilluminator is not really good for the user's health because you would be exposed to the UV light as well. It does come with a glass cover on top and you will be wearing protective gear, but it is always better to use a chemidog if you simply want to visualize the gel, because this will ensure that you are as far away from the UV light as possible. However, if you want to extract this DNA from the gel, because that is something that you can do when you have separated, like say for instance here, take a look at this lane. So it has multiple populations, but the beauty of agarose gel electrophoresis is that not only does it allow you to separate out these different populations on the basis of its size, you can also excise out your population of interest from the gel. So you have a mixture and of DNA molecules and you want only one type of DNA molecule from the mixture. So what you can do is you can run an agarose gel and you can excise your DNA of interest from the gel. So what would you do over there? Because this gel is pretty well, it is, I will not say it's brittle, but it can break pretty easily. So you take a sharp blade and you cut out a box which contains your fragment of interest. But you have to ensure that when cutting out the box and when, so what do I mean by cutting out the box? So let's say this is the DNA fragment you want to isolate. So you take a sharp blade and you cut a rectangle around this particular fragment. So when cutting this rectangle, the UV light has to be on, otherwise you can't really see where you're cutting this. And when cutting this rectangle out, you have to be careful to ensure that you do not cut within it. Because if you cut within it, then you will be cutting through the DNA molecule and this will shear or fragment your DNA molecule. So that is also undesirable. So you simply cut around it and you leave some extra agarose around the edges, because with the help of that extra agarose you're going to simply or how you're going to simply pick out this block of gel containing your DNA of interest, because you will like just stick the blade into this extra agarose at the edge and pick it out, or pick out this agarose block out from the entire gel. So that is all about DNA isolation and visualization. So to briefly recapitulate, we talked about a step-by-step procedure of genomic DNA isolation. We talked about isolation of genomic DNA from animal cells, but the steps would be the same if you're going to isolate DNA from plant cells. The only point of difference would be in the first step of cell lysis. The rest of the steps would remain the same. So we talked about the different steps. We talked about the underlying principle behind each of the steps. We talked about why we are using whatever reagent we are using, what function or utility it serves. Next, we talked about visualization of DNA, and for that, we discussed agarose gel electrophoresis. We talked about the principle of agarose gel electrophoresis, the different factors that are affecting the rate of migration of DNA through an agarose gel, and how to cast an agarose gel. We also talked about visualization of an agarose gel. So this brings us to the end of this topic of DNA isolation. And if you have any questions, please feel free to leave it in the comment section and I will definitely get back to you. In the subsequent lectures, we'll be talking about some of the other recombinant DNA technologies that we will perform once we have isolated the DNA. But that is something we will discuss later. So let us not bring it up over here. If you like this video, then do like and share it. And if you are looking forward to more such content on different laboratory techniques, then do subscribe to the channel. And I will see you all next time. Bye.